from Microbe TV. This is Office Hours for Wednesday, July 3rd, 2024. I'm Vincent Racaniello, and welcome to your viral corner of the internet. Here is the place where you can come and ask virology questions of a virologist. And in my opinion, expertise is still useful. Much of the U.S. apparently now thinks it's not. Okay. You can ask virology questions. So what we have for you tonight, you can ask your questions. I'm going to do a mini lecture. And of course, we're going to find out where all of you are from. So cue up your I'm from, and I will highlight it and bring it here into the stream. Meanwhile, thank you to our moderators for joining us. Les is here. Andrew is here. Les is from California. Andrew is from New Zealand. Barb Mack is from the UK. Tom is from the Oregon Coast Range. Who else do we have tonight? That's all I see tonight. If someone else comes, thank you very much for moderating, and thanks everyone for coming tonight. Where are you from? Let's see. Will is from Changsha, China, where it is sunny and 35. By the way, I'm in New Jersey tonight, where it is 8 p.m. It's sunny and 25 Celsius. And looks like it's going to be warm the rest of the week. Nice weather. Changsha, China. I know Abdul Aziz is from Saudi Arabia. Looks like Barb Mack is in Worcestershire. Lise is from Columbus, Ohio. Stop raining successfully repaired the garbage disposal. What more can you want from a single day, right? Yeah, I posted today's live stream late. I'm sorry. But we're here, and it's on the schedule, which is at microbe.tv/calendar if you'd like to know. I try and keep that up to date. Where else are we from? Patricia is in D.C., where another heat wave is coming. Mark Martin, Tacoma, Washington, University of Puget Sound. By the way, hit the like button. There's only 50 of you so far, but hit the like button. Why not? Get more people here. So less like the Beyond the Noise 40 shorter SARS-2 origins theme in the discussion and rapport. Yeah, when something's important, you cannot do it too much, right? So Paul said, I'd like to do this. I said, sure. Yep. Kang is from Chicago, 33C and partly cloudy. Senator William Proxmire of Wisconsin gave the Golden Fleece Awards. Correct. He picked out, I believe, NIH-funded research that he thought was absurd. And of course, he had no clue what was absurd because he's not a scientist. The paper we did last week on TWIV, where they knocked down prion levels with an adeno-associated virus vector. Oh my gosh, that whole thing is based on decades of fundamental basic science research and discovery. No one set out to do that. They didn't set out to knock down prion proteins 40 years ago. They set out to say, look at these two proteins. They can come together. What's that? Turns out to be protein splicing. Gene expression can be modulated by changing the methylation state of the DNA. Let's explore. There's a methylase and there are other proteins. People just get curious. And the Congress people who are now in line to control the purse strings don't get it. They think you have to cure cancer, you have to cure diabetes, you have to cure hypertension. Okay, that's fine. But you need at least 50% of people just following their curiosity. Now, maybe you're tired of me saying this because I say it every week, but you can't say it enough. Serendipity drives science. And so Proxmire thought, working on Worms. It's not a good idea, but we learn a lot of fundamental things from worms. Oh my gosh. Of course, bad science we don't want to fund, but curiosity we do. We do. Mark Martin has sent me some gifts. I will show them to you because they're pretty cool. As soon as we get through where people are from, Questella is from Ohio. I know that. Pete is in London. It's cool. And yes, of course, Michael Schmidt talked to Mark Martin about copper. It's great. Yes, Proxmire was a good, honest senator, but the Golden Fleece Awards were an embarrassment. Absolutely, because he doesn't know any science. Didn't know any science. There's some. Let's see who else is here. Brian's from Montreal, and Wayne is from Urbana, Illinois. Don't worry is from San Francisco. Rima is from Iowa. Claire is from the UK. Red is from Idaho. Well, yeah, you could take care of yourself, Red, right? Little Vinny is hysterical. Yes. Little Vinny is the name of the iPhone, the official incubator iPhone. We decided not to get a landline. Who gets landline these days? And instead, we got an iPhone. We call it Little Vinny. Yeah. Joseph is from Ontario. Jan is from Tucson, Arizona. And Les, I told you, is in California. Is 98F. Wow. 37C. Spar is from Boston. Maureen is from Northeast Ohio. Abdur is back in New York. Karen's playlist, North Maine, 72F. Very good. Mark says he's still hoping to get Novavax at some point. I'm pretty much Moderna so far. So now many of us are beginning to think that perhaps the reason you get reinfected so soon after you're vaccinated is because the mRNA vaccine immunity is not durable. Maybe protein-based vaccines would be. So there's no long-term studies of Novavax, but many people are now telling me, including someone I talked to in Stockholm a couple of weeks ago, Novavax, protein-based. Look how good Shingrix is. Protein-based vaccines. So look, I don't want to denigrate mRNA vaccines. They were useful in an emergency. But I think for long-term immunity, they may not be what we want. S is from Vancouver, BC. Kip and Laura, here we are from San Francisco. Touj got tested positive for COVID second time. Hat is from the LA suburbs, 106. Wow. Jill is from Wilmington, Delaware. Jonathan is from Encinitas. I looked at the calendar a minute before, and July, August is not on the calendar. Let's take a look. microbe.tv/calendar. Well, first of all, it's our calendar, I think. Yeah, why didn't that show up? May. You're right. I'll have to fix it. Another sticky note. Everything has gone the way of sticky notes. You know, we're here on office hours, we tried a lot of things over the years to remind me to do stuff. And in the end, I'm going to put a sticky note. Update microbe TV calendar. I'm sorry about that. Here I am boasting that you could simply go to the calendar, and I was wrong. You know, I will admit when I'm wrong. Absolutely. And that's one of the many times that I am wrong. Thank you very much, Pete. Thoughts on getting Novavax? Well, I think you'd need more than one, right? Because that's how you get the durable memory. It's not clear to me that mRNA and then Novavax would give you that durable memory. I mean, these are interesting questions, but we don't have the data to address them. So until then, get multiple Novavax, especially if now, you know, there's a new Novavax matching the circulating variant. Just get a couple of courses of that. But I'm not giving you advice. Go listen to Daniel Griffin. I'm talking from an immunological perspective. One would need more than one dose to generate memory to the protein-based vaccine. MK is from Eastern Massachusetts. All right, Tuj got tested positive, had 3 vaccines. The second time, 2 years from the first infection, had the same symptoms, mild, a lot of coughing. So I think it's the durability is not good. There's not good immune memory from these mRNA vaccines. So that's why you're getting you're going to get reinfected anyway. But disease should be prevented by T cells, at least severe disease. And I think even memory T cells are not being made by the mRNA vaccines. But I'm speculating because the studies haven't been done. Mike and Lori are from Seattle, Washington. And Jan says we have learned more from C. elegans than almost anything else. Well, not almost anything else, I would say, but a lot. And yes, people say, why do we have to work on worms or flies? Idiots. Where's this country going to? Every day I'm disappointed in this country. Look at the political mess that we are in. The Supreme Court. You know, I know there are people here who are conservative. That's fine. But the stuff they're doing is ridiculous. They're messing with science too. Oh my God. S. Wes is in Vancouver, BC. And there'd be a discussion on LV et al. Paper on SARS-CoV-2 early. Yeah, I did. I said that last time. I'll make another sticky note. LV et al. CoV-2 early lineages. You know, in general, if a paper isn't strong, convincing, we don't do it on Twitter. We don't give attention to subpar science. Why? Well, there's only a limited amount of time that we have, and we want to cover good science. How do we know? Well, we got a lot of virology expertise on the panel. And you know, some of the skeptics would say, "Whoa, you think you know because you're biased, you're beholden to big pharma." This is all crap. I'm not beholden to anyone. I'm beholden to truth, correct science, okay? It's ridiculous to say stuff like that. So yes, if it's a good paper, we'll do it. Yeah, politicians think cancer is one disease. Yeah, I mean, they also don't know what gain of function is, right? They want to ban it. They think it led to the current pandemic, so they want to ban it. This is so ignorant, it burns my brain. I can't function. I have to do other things because I can't handle the stupidity. Ay, ay, ay. Abdur says, if we do something on the scope of the Human Genome Project, maybe we can knock some of the larger diseases off. I don't think so. We're not even going to knock polio off. Eradication is not happening. I got news for you. After X number of years, 35 years, not happening. So I don't know about other diseases. We got lucky with smallpox. Zan is from New South Wales. Nebby is from Central Florida. Fields of Green is from Salinas Valley, California. Veronika Privyet is from Moscow. Privyet. I have just a few Russian words that I'm learning. Of course, I have Privyet, and I have Spasibo, right? I have Poka and Dozavtra. 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 Welcome. Veronika. Logic is from Chicago. Peter is from Boulder, Colorado. Yeah, you need both serendipity and directed research. Didn't I say 50%? Pete Glass? Yep. Theodore is from Athens, Greece. Cool. Greece. Science 2 is from a Native American reservation, half in Idaho, half in Nevada. Well, welcome. I have not seen you before here. John is from Minnesota, Minneapolis. And I know that it's July 3rd. It's the eve of a holiday for many U.S. people, but we're still here because science does not stop. N.Y. Cohen is from White Plains, New York. Patricia's from Massachusetts. Twin Daddy from Minnesota. My volume is extremely low. Does everyone agree? I can crank up my volume a little bit. Yes, 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 yes. When I get angry, my blood pressure goes up. Yeah, people tell me I should stay calm because it's bad for my health. I don't know what to do because I don't have a system for calming myself. My dad used to get very anxious. He was born in Italy, so he had the over the top genes. He'd get upset at everything, and everyone was scared of him. So no one, told him to chill it. Apparently, I've inherited that, but I don't do it for everything. I just do it for things that mean something to me. Mm. Thank you very much, Veronica. I tried to pronounce other languages well. Your public is with you. Yes, thank you very much. I appreciate it. My limited public is with me, but we have to get the other people on board, right? The 2/3 of Americans who think this COVID-2 came from a lab. It's ridiculous. It's totally ridiculous. And then if you try and talk to them, they don't want to listen. another uh, praise for my Russian skills. I only know 3 words, so, but I try and pronounce properly, right? Dave is from Georgia. Environmental Coffee House, somewhere upstate New York, if I remember. It's hard for us New Jersey Italians to stay calm in the face of all the crap that's going on in this country. Congress, Supreme Court, they make decisions which are just contrary to logic. It's what they want, but it's contrary to logic. So let's get rid of government and we'll have a king who can get away with anything. Doesn't that make sense? Gosh. I cannot be in the company of others because I just fume. So I'm not going to do this tonight. Use breathing to calm you down. I heard that works, but now I have to talk and give you information, and then that's where we get into trouble again. Go bird watching or fishing, take a walk in the garden or the woods. I can't do that now, right? I'm here with you for a couple of hours, and I'm really happy to be here. And then when I'm done at 10 p.m., I'm not going to take a walk. I think many people on this channel are with me, like Environmental Coffee House. I'm as angry as you are. I've been making videos on my channel all about it. It's crazy. Yeah, cats should rule. Maybe if cats ruled, everything would be good. All right, let me. I'm going to show you some. So Mark Martin is a nice man. He's always grateful, and he sends me gifts all the time. So I'm very grateful. So he sent me 3 gifts recently, and I'm going to show them to you. This is a bag of viruses. So these are all different colors. Nice crinkly. You hear it? What do they call that noise that you crinkle and you fall asleep? This is a good one, isn't it? Someone tell me the name of that that noise. Anyway, so it's full of different colored, obviously viruses, right? And Mark says I should throw them at people who make me angry. Unfortunately, I don't have autofocus on this camera. Sorry about that. But I can put it, here's the plane of focus. And then I get a cramp in my freaking hand. Isn't it cool? So, well, it's got a little plug here. I'm not going to open it up because then some liquid will spill all over my keyboard. But it's squishy. And look what happens when you squish it. You push it. A little thing pops out. And you can see there's some kind of gel inside. Yeah, little like tapioca balls, but yeah, little tapioca balls, but not. And then there's some liquid in. See, I'm analyzing this structure, but it's kind of cool. I'm going to bring a bunch into the incubator and put them on the shelf. Okay, so thank you. That's very cool, Mark. I like it. And next thing um, is a virus kit. It's pretty cool, actually. It's here, cell and microbiology models. So they make a bunch of different ones. So this is virus. Okay, so what we have here, I'm sitting on this wire, it's pulling, and I'm going to get annoyed. Let me just fix it. I don't really need earbuds today because there's nobody else to hear, but I like hearing myself. Okay, so this model comes with instructions, okay? But I don't need no stinking instructions because I know you start with the capsid. In this case, it's going to be a nucleocapsid. So here's the nucleic acid, okay? Now, those of you in the know may think this looks like an HIV core, because it's got this conical shape. So it could be, but they don't say. Anyway, you put that in here, okay, and then you put the capsid together. I'm not showing you properly. Put the capsid together. Now this is not fitting perfectly, but now we have a capsid. If this were the virus, by the way, this is a good teaching moment. If this were the virus, it would be a capsid. Can you name a virus that looks like this? Nucleic acid inside with just a naked capsid. Poliovirus, adenovirus. It's a capsid. Anyway, this kit comes with an envelope, okay? And so then you assemble the envelope around the particle. Let's see if we can do that here. What the hell? I think these things have to go in a certain way. Well, maybe I should make half first. Half first. It's a little imprecise, but it's okay. It works. So we got that, and now we put our nucleocapsid inside, right? Isn't it pretty? I'm going to make the other half first. And you can see, of course, that there are spikes in this envelope. And tell you why it's not HIV based on the spikes in a moment. So now let's take these two halves and put them together. There you go. Now we have our envelope virus. It's clear and has spikes. Beautiful spikes. I like these very much. Oops. Fell apart. You don't want to glue this, of course, because you want to be able to take it apart and show people. This is a great teaching tool, Mark. But we have two different kinds of spikes in the envelope. The envelope is this clear plastic shell, right? And inside is the nucleocapsid. And then we have two different kinds of spikes. We have these don't line up right, but it's okay. You get the idea. We have the red ones, which are very pretty. They're a little bit weird because most spikes are trimers, and this has no obvious symmetry, okay? So the people who made it didn't know what they were doing. And then there's another kind of spike here, smaller, right? So this is not HIV because HIV has one kind of spike. But these are sparse on the particle, which is like HIV. So these bigger ones, if we're just those, I say it was HIV because they're very sparse. They're not densely packed as for influenza viruses. So I don't know what virus this is. And it doesn't say here. Let's see. Inos virus. Yeah, it doesn't say what they think it is. Booklet and model instructions. Let's see what they say in the instructions here. Okay, they're teaching me a little biology here. I don't really need that. Bacteria, viruses. Last page. Viruses are not cells at all. They are limited organisms that contain genetic material. They have no cytoplasm. They're quite dormant by themselves. They need to infect the cell. So they don't tell you what this might be. This is just a generic virus. I really like it, though, with all my criticisms. I really like it. Thank you, Mark. It will become part of the incubator legacy. If you find things like this, I mean, I don't know where Mark finds this stuff. This is just cool. Cell and microbiology models. Buy it and check it out. They're not very expensive, right? Okay, so that's 2. And then the third one is just very nice. You're going to love this. It's a glass virus. Oh, my God. It's red. It matches my glasses tonight. Holy crap. Look. Yeah, they can call me mad. It's fine. Isn't it? I put it here, but you're just going to see a mush. Oh, my gosh. It's very beautiful. It's a solid glass ball with spikes. So it's hand blown, obviously. And this is made by a company called Green Glass. I have a shop on Etsy. Let's see. Green Glass, which is etsy.com/shop/Isabel Green. So the lady's name is Isabel Green. She's a glass blower out of Cape Cod, Massachusetts. Graduated from Falmouth High School in 2008. Worked at a glass studio for 15 years and then went out on her own. This is gorgeous. Now, all 3 of these things are lovely, Mark. This is particularly gorgeous. This will go to the incubator as well. It's going to be behind me. I love it. I really like this. It's a little tinted red to match my glasses. So thank you very much. Beautiful. Thank you, Isabel Green, for making this. Okay. I love this. You can send me stuff and I'll play with it and explain, right? If you want. Pay for it if you want. I like to get models and explain all about them. I'm going to do a little I'm going to do a little mini lecture. I need to do it now because last week I said I'll do it later and then it got too late because we got to talking about all kinds of things like milk. So let's do a mini lecture. All right. So let's see if I can remember how to do this. We turn on screen sharing. We have to make this slide thingy very big. Otherwise people can't see it. Okay. It wouldn't take long. Would take long. I'm going to wait for the stream to catch up because I'm looking at the stream, which is delayed over there. Just want to make sure it's okay. Make it a little bigger, maybe. It's at 54.75 is going to be too big. Yeah, it's not bad. I think that's good. Okay, I want to talk about therapeutic viruses. This is the last lecture of the virology course. And it's like a culmination of the course because everything you've learned in the course now gets put into play in making viruses to cure diseases. So last week on Twitter, and you really need to go listen to that episode, we talked about using adeno-associated viruses to knock down prion protein levels. We had a little mini lecture on prions, remember, not too long ago. These are misfolded proteins of our own that cause fatal neurological diseases. No cure, but it turns out if you knock down the protein levels, you can reverse many of the symptoms. They used the technology we're going to talk about today to deliver a gene editing, actually a methylation machinery to the promoter for the prion gene and turn it off. Anyway, so therapeutic viruses means a couple of things. First, you can use phages to treat bacterial infections, of course. You just talk a little bit about that today. You can do gene therapy. You can deliver a gene to patients who lack the gene or who have defective versions. You can deliver antigens. You can make vaccines using viral vectors. So during the pandemic, we had a few like adenovirus vectored vaccines famously. And finally, viral oncotherapy, treating tumors or cancers with viruses. So 4 applications of therapeutic viruses that I want to talk briefly about today. Now, all of these depend on making an infectious DNA copy of your viral DNA. And I will illustrate this with poliovirus, but it can be done with every other virus now. This experiment, by the way, I did as a postdoc in 1981 with David Baltimore. It was the first infectious DNA of an animal virus. It is arguably why I got my job at Columbia, for sure, because it was an amazing finding. And I did it not because I wanted to develop the field of gene therapy to cure diseases. I wanted to see if it could be done because I knew it would open up many possibilities. I was just curious. Actually, David was curious. He told me to do it. I wasn't smart enough to come up with that. So you take your virus. In this case, it's poliovirus. The genome is RNA. You extract it. If you put that RNA into cells, the cells will make new virus. The RNA can get into cells and initiate reproduction cycle. But if you make a DNA copy of the RNA using reverse transcriptase, you put it in a plasmid, which you can propagate in bacteria. You can put that plasmid into DNA, into cells, and the cells will make virus. Or you can do in vitro RNA synthesis, make RNA and put the RNAs into cells, because the RNAs are more infectious than DNA. And by the way, that's the same technology that was used for the mRNA vaccines, taking a DNA copy of the spike gene and transcribing in vitro to make RNA, which was part of the vaccine. So this now can be done with every virus. The key is you can modify DNA. You can add bases. You can subtract bases. You can add genes. You could remove genes. Basically, it enables you to do the things that we're going to talk about, today, to make therapeutic viruses. Now, first, phage therapy, very briefly, because we've talked about this extensively on many programs. Darrell discovered phages in 1915, and he thought they would be useful to treat bacterial infections. He co-founded the Eliava Institute in Georgia, which is active to this day. That institute produced phages for antibacterial therapy during World War II. Many of the soldier kits have vials of phages in them. But of course, the introduction of antimicrobial drugs in the 1930s dampened enthusiasm because here was an antibiotic. It was so easy and cheap to make. So really, in most places, although the Eliava Institute continued its research on phage therapy, but now, of course, we have widespread resistance to antimicrobial drugs. So it's revitalized interest in using phages. And you've all heard about this. The principles are you use a lytic bacteriophage, a bacteriophage that kills its host to kill a specific host. And it has to be specific. You don't want to kill your normal flora, for example. And so this complicates matters because you need to, if someone has an infection that you want to treat, you have to identify the bacterium and find a phage that will kill it. It takes a little bit of time. So that's the approach. But lysins, the proteins encoded in phage genomes that lyse the bacteria, have also been exploited. They have been used for surface decontamination. And then we have two phages approved for plant applications, one to treat bacterial tomato canker and then another to treat listeria, which is a common contaminant of meat and poultry. So, and as you know, you have heard Stephanie Strathdy tell the story of her husband who received phage therapy for his infection, and it saved his life, apparently. And that's right here on this slide. This is development and use of a personalized bacteriophage-based therapeutic cocktail to treat a patient with a disseminated resistant Acinetobacter baumannii infection. And so there's Stephanie Strathdy here somewhere. There she is. Because Forrest Rower is there. And she said to the docs taking care of her husband, what can we do? They said, look into phage therapy. So she Googled it. And these phages were discovered by the Navy as well as by Ry Young's group at Texas A&M. And we visited them for TWIV 502, and we did a TWIV about this with some of the principles. But we have a couple of other articles here on phage therapy. Here's one from Paul Turner, phage treatment of an aortic graft infected with Pseudomonas aeruginosa. This is an interesting case of a patient whose graft became infected, and they simply opened him up and poured phage on it, and it cured the infection. We talked about that on TWEVO 44. And then here's another one from Graham Hatfull. Engineered phage is for treatment of a patient with disseminated drug-resistant Mycobacterium abscessus. So, it's very early days, but we're moving forward in the treatment of infections with bacteriophages. I did a session at South by Southwest back in March with Stephanie Strathdee and another guest where we talked about this. So you might want to go and check that out. There's you can just search for phage therapy South by Southwest. You should find it. Okay, that's all for phage therapy. Let's talk about virus vectors now for other purposes. There are a number of vectors that have been developed. One of them is adenovirus, right? It's a large DNA virus with a double-stranded DNA genome, about 36,000 base pairs of double-stranded DNA. And so a virus is highly manipulable. We can cut genes out, and we can put other genes in. And so the first generation adenovirus vectors, which were made many years ago, were deleted in two genes called E1 and E3. And E1 encodes what are called T antigens. These are proteins that counter two cell checkpoint proteins that regulate the cell cycle. And so you can provide those in a cell line. So the virus need not have it, because when you give people the vector, it doesn't have to replicate. It just has to get into the cell and deliver the DNA. And E3 encodes immunomodulatory proteins, so it's not essential, and that can be deleted as well. So that gives you a little room for inserting genes. But then they made second generation with E1 and E3, but also E2 and E4 deleted, giving you more space for the transgene. Just, by the way, a diagram of the virus genome, of course. And now we can make third generation gutless vectors, which have nothing but the two ends, the very two ends, the inverted terminal repeat. There's two repeat sequences at either end, and a packaging sequence. And you take everything else out, you have packaging sequences, and then you stick your transgene in. So this is what the gutless vector looks like here. ITR, packaging sequence, transgene with a promoter, and the other ITR. So you can put quite large payloads in this vector. You can grow it. You can make very high quality preparations, very pure, and these can be given to patients for various indications, which we'll talk about in a bit. But I want to go through vectors first. Adeno-associated virus is also very popular. Now adenovirus is a lytic infection. So it goes through a cycle, and that's the end of it. But adeno-associated virus persists in the cell for many, many years. And so that's a really favorable property, right? It's a single-stranded DNA virus, a relatively small genome. You can see 4.6 kb here, packaged in a icosahedral capsid. Only encodes a couple of proteins, a replication set of proteins, in the capsid. And so to make this into a vector, what we do is we take out the replication and the capsid encoding proteins. We just have the inverted terminal repeats. We put a promoter on the left, and then we put a transgene in. All right, so that is the carrier. So then we have to package that into a virus particle. You put that into cells along with another plasmid that encodes the rep and the capsid plasmid, so that you can get this DNA replicating and you can get it packaged. And then you need other functions. This is a defective virus, or it's a helper-dependent virus. It's dependent on adenovirus. So there's a number of functions from adenovirus that you put in as well. But you get out a recombinant AAV that have this genome with the transgene and nothing else. So when this goes into cells, it will not replicate, but it will make the protein. Very cool. And you get long-term expression. There are multiple serotypes. You can engineer this virus. Excuse me. You can engineer the virus so that it will infect different tissues. It's like neurotropic and muscle tropic, liver tropic, heart, and so forth. It's beautiful. And again, years ago, a few labs started working on AAV. They thought this could have interesting applications, so let's figure out how it works. Curiosity. We use retroviruses as vectors. With retroviruses, you can take two plasmids, one encoding the GAG-POL gene, which is basically the structural and the enzyme proteins of the virus, and then you put the envelope or the spike on a separate plasmid. You put both of these plasmids into cells, and out come empty particles. There's no genome here, so this is an empty retrovirus particle. It's really cool that this GAG-POL and the envelope were enough to drive production of particles. Really amazing. All right, so now you want to put a gene in. You add a third plasmid. It's called the transgene plasmid. You put your transgene in with a promoter and also a packaging sequence on that gene, so it will get packaged into the particle. So now we make retroviruses with whatever your foreign gene is. And the advantage of retroviruses, of course, is that their RNA is converted to DNA and it integrates into the cell DNA, so making expression forever. So mainly these days we use retroviruses based on lentiviruses like HIV-1, but there's some other retroviruses that are used. This is amazing to me. We take a virus that has killed over 35 million people, and we tame it to use as a vector. Just leave scientists alone, okay, politicians? Some of the advantages of HIV can infect non-dividing cells. You get long-term expression because the DNA integrates. That's called a provirus. You can put up to 8 kb in these particles. However, these vectors can insert in bad places in the DNA of the host. So they have modified the vectors over the years to take care of that problem, that potential problem. Pox viruses are also used as vectors. Some experimental COVID vaccines were derived from pox viruses. The one that's used is called modified vaccinia virus Ankara. It was originally made as an alternative smallpox vaccine. In fact, it's part of the U.S. Strategic National Stockpile. It was used during the Mpox outbreak. The vector is infectious in avian cells, but not in mammalian cells. It was made by passaging the virus in chicken cells until it's sustained an assembly block. So you can infect mammalian cells, it will deliver the DNA, it will get expressed, but it will not make new virus particles. This is a very safe virus to work with. BSL-1, the lowest laboratory safety conditions, has a large capacity, and we also have a canary pox virus that has been modified similarly. Now, how you put genes into this is very interesting. You have your MVA, and you take a plasmid where you have your transgene in the plasmid, and then the transgene is here in red, and then you flank the transgene by sequences from the virus itself. These are called recombination sequences. They're going to make this plasmid recombine with the viral DNA. So you put that DNA in cells you infect with MVA. The DNAs recombine, and out comes MVA with your transgene in it, which you can then use. And just one more. We have more virus vectors that we use, but I just want to tell you one more, VSV, vesicular stomatitis virus vectors. These, of course, are negative strand RNA viruses. When they infect the cells, they produce multiple mRNAs. And so what you do is you can replace the glycoprotein of VSV with another glycoprotein of interest, a transgene. And the way you do that is you take 4 plasmids encoding the viral proteins NPL. You take your transgene plasmid, you put them in cells, you drive the synthesis of mRNA using a T7 encoding vaccinia virus, and out come recombinants with a viral glycoprotein. So this was experimentally attempted to make spike SARS-CoV-2 vaccines, but it didn't work out. But it's also used to do neutralization assays in labs that don't have the facilities to work with infectious SARS-CoV-2. Okay, let's talk about some of the licensed vaccines that use viral vectors. There's the Zaire Ebola virus vector. Dengvaxia is using a yellow fever vaccine vector. This has also been used for a Japanese encephalitis virus vector and a West Nile virus vaccine, for horses. A Newcastle disease virus vector, which we didn't talk about, is a vaccine for H5 avian influenza virus for chickens. Of course, the Ad26 vector was used for the Janssen COVID-19 vaccine. This is no longer used because it was associated with severe blood clots. Chadox vector was for SARS-CoV-2 by AstraZeneca. And then Sputnik, made in Russia. Ad5 and Ad26 comprise the Sputnik vaccine for SARS-CoV-2 made by Gamaleya. So these are just some of the many. And of course, as we mentioned before, mRNA vaccines really took the forefront and did the bulk of the work in many countries for controlling the pandemic. And remember, mRNA vaccines are a small piece of RNA encapsulated in a lipid nanoparticle. These are taken up by cells. The protein is made, and then it's presented to antigen-presenting cells to initiate an immune response. So you have to put a lot of mRNA in you, right? Many micrograms. What people have developed now, and which was talked about on a recent TWIV, is self-amplifying RNA. So again, you have an RNA in a lipid nanoparticle which is injected, but this RNA not only encodes your antigen, say a spike protein, but it also encodes a polymerase that will self-amplify. So you can start with very, very much less mRNA, and it will self-amplify. Now, whether this ever gets traction, one was actually licensed recently in Japan. Whether it gets traction is a good question because I don't know if it's going to be durable, right? Like we talked about earlier, it's mRNA based. Maybe protein is the way to go, but I just want to point that out as an interesting technology. All right, so now 2 applications: gene therapy for monogenic diseases. That is, you have a mutation in one gene, which happens in 1 out of 200 live births globally. There are over 6,000 different monogenic diseases, and these are amenable to viral gene therapy. You can give people the correct gene in a viral vector. And there are over 1,800 clinical trials ongoing for these kinds of monogenic diseases. Here's just a few of them. For example, SCID, severe combined immunodeficiency. Here's the defect and the incidence is being treated with retroviral vectors, lipoprotein lipases, hemophilia, hemoglobinopathies. Here's some blindness conditions, which we will talk about, and others. So these are just a few of the many, many, many monogenic diseases that are amenable to gene therapy. And in general, what you do, there are 2 ways that you can deliver the gene to patients. So you can make your virus with the gene using the vectors we've talked about. You put the gene in and you simply inject it into the patient. Or, and then when you do that, most of it's going to go to the liver because the liver clears things that are in the blood very rapidly. And if you want to target the liver, that's great, but often you don't. So the other approach is cell-based delivery. And there are 2 ways of doing that here. In this case, we take T cells from the patient and we infect them with a virus that now synthesizes a new chimeric antigen receptor, which is targeting, say, a cancer or a tumor protein. And so we put those cells back into the patient. Or we can take stem cells from the patient and inject the vector into them, make sure the stem cells are making the protein, and infuse those back into the patient. So the advantage is you're putting cells in. They will presumably populate many different locations in the patient. So let me give you a couple of examples. There are a number of diseases that cause blindness. These are called inherited retinopathies. They're common, untreatable blinding conditions. They're monogenic. They typically comprise mutations in retinal photoreceptors and retinal pigment epithelium. So here, at the back of the eye, right? The the retina, and that is composed of the retinal pigment epithelium and then a series of neurons, remember cones and rods and Mueller cells that eventually synapse with the ganglia and transmit. And so um, there are a number of genes in the retinal pigment epithelium, in the cones and rods, in the Mueller cells, and the ganglia themselves. All those mutations are identified here, and they're being treated with AAV vectors, lentivirus vectors, and adenovirus vectors. The one that has made the most progress is now licensed to treat Leber congenital amaurosis. So it's mutations in RPE65, which is one of the genes in the retinal pigment epithelium that's defective and causes blindness. It encodes a protein required for photoreceptor function. In dogs, it was shown that a single subretinal injection of an AAV vector with the canine RPE65 gene restores visual function, and it lasted for years. And this was then tested in patients. I discussed it with Catherine High from Penn, who had been involved in the development and clinical trial of this vector. And it was approved in December 2017. It's called Luxturna. First gene therapy approved in the U.S. to target a disease caused by mutations in a specific gene. It works. For two eyes, it'll cost you nearly $1 million, $895,000. So none of this is cheap. Some other gene therapy trial successes. SCID can be treated with a lentivirus delivering the IL-2 receptor gamma chain gene. Cerebral adrenoleukodystrophy is delivered. The gene is defective, is ABCD1, and the wild type is delivered in a lentivirus. That's a $3 million treatment. And then sickle cell disease is treatable with two different products, Lyfgenia, which is a lentivirus carrying the beta globin wild type, and Casgevy is a virus delivering CRISPR-Cas that will edit out the defective gene and put a new one in. Beta thalassemia is also treated with Zynteglo, a beta globin in a lentivirus vector. Hemophilia A and B, you deliver clotting factors 7 and 9 respectively in an AAV vector. That's $3 million a pop. Lipoprotein lipase deficiency, you deliver the lipoprotein lipase gene in an AAV vector, $1 million. Spinal muscular atrophy, you deliver the gene in an AAV vector, $2.125 million. And Duchenne muscular dystrophy, you deliver the DMD gene in an AAV vector. So you see increasing numbers of gene therapy successes, and this will only increase. It's very interesting. All right, finally, you can treat cancers with viruses, viral oncotherapy. You destroy tumors with viruses. And in general, there are two approaches. Some non-human animal viruses selectively replicate in human tumors, like myxoma virus, a pox virus, Seneca Valley virus, a picornavirus of cows or pigs. They're not human viruses, but they replicate in human tumors. People just looked to see, and they found that they could kill the tumors. Or we modify viruses to target and kill tumors, and usually we have immune enhancement. There's a lot of modification going on in genetic modification of these vectors. And Linda Coughlin, some time ago, we used to do a podcast called Virus Watch. She talked about these. You can find that on microbe.tv. Now, one of the things that's taken advantage of in using viruses to kill tumors is that cancer cells often have interferon defects. All right. So cancer cells don't make interferon and other cells do. So you can take from the virus that you're going to use, you can take out the genes that antagonize interferon. And that will restrict the virus to reproducing in the tumor because there's no interferon response in the tumor, but there is in other cells. So if the virus gets somewhere else, it's going to be restricted by interferon because you've removed from the virus genome the genes that antagonize interferon. So this is one of the brilliant outcomes of fundamental research. People said, oh, this virus antagonizes interferon. Let's see how it works. They didn't say, we're going to make a cancer-killing virus. No, they just wanted to see how it works. You can modify the virus to make it tumor cell selective. And we can do that by taking out the interferon antagonism genes, as I told you. It turns out that the measles virus vaccine binds a different receptor from the wild type, and that receptor is displayed on many tumor cells. You can engineer the glycoprotein of herpes simplex virus to contain proteins that will target it to tumors like gliomas and breast tumors. You can do the same with adenovirus. You can insert domains that recognize tumor antigens into the virus particle. Basically, you can modify viruses in ways that make them tumor cell selective. You can also do post-entry targeting. For example, you can make your virus, your cancer-killing virus, using a promoter that doesn't work in a healthy cell. And only when it gets into a tumor cell will the promoter work and make whatever protein that's going to be anti-tumor. You can also put the targets of microRNAs into your vector for microRNAs that are present in normal cells but not in tumor cells. So microRNAs will bind the viral mRNAs and inhibit the virus reproduction in a normal cell but not in a tumor cell. And then we can do other strategies for increasing cell lysis. For example, we can put the pro-apoptotic protein p53 into adenoviruses. We can incorporate cell genes that are pro-drug convertases. So, for example, we can put acyclovir into, we can put HSV-TK, thymidine kinase, which will convert acyclovir into a chain terminator. We can do this with other drugs as well. And then we can put genes for viral proteins that allow cell-to-cell spread and shield the viruses from antibodies. And so here's another example of arming with pro-drug convertases. So we put thymidine kinase into a virus vector. It converts ganciclovir to ganciclovir triphosphate, and that will cause chain termination of DNA. We can put cytosine deaminase in, which will convert 5-fluorocytosine to 5-fluorouracil, and these analogs stop DNA replication of tumor cells. So the virus is infected a tumor cell. It's making these pro-drug convertases, and then you give the patient the drug separately, and it gets converted into a toxic metabolite. And that's the basis for TOCA 511, which is a retrovirus armed with cytosine deaminase. It's given intratumorally along with 5-FU. So we have a tumor in the brain, you inject the virus into the brain, and then you give TOCA or 5-fluorocytosine orally. It's converted to 5-FU, and that presumably will destroy the tumor by inhibiting its DNA synthesis. The other thing we do is promote anti-tumor immune responses. The tumor microenvironment is immunosuppressive. Both innate and adaptive responses are disrupted. So we try to overcome that. We put cell surface proteins for checkpoint inhibitor ligands that block T-cell responses. We can do monoclonal therapy plus an oncolytic virus or even incorporate the monoclonal antibody gene into a virus. So in tumor cells, these T cells have problems killing the cells because of checkpoint inhibitors, and we reverse those with the vectors. We can modify the genomes of many viruses to remove genes that block type 1 interferon, as I said earlier. And we can also include immunostimulatory proteins like GM-CSF that will be made in the tumor and recruit immune cells to work on it. And in fact, herpes virus vector has recently been approved for treatment of melanoma. So this is a virus called telimogene laherparivec, or TVEC. Into the herpes genome is the gene for GM-CSF that stimulates the production of granulocytes and macrophages that stimulates adaptive immunity against tumor antigens. They've deleted 2 genes that cause tumor-specific replication. They delete a viral gene that doesn't inhibit, so that the virus infection will not inhibit antigen presentation, so T cells will lyse the tumor cells. And this went through a phase 3 and was licensed in 2015 for the treatment of melanoma. We now understand that the therapeutic efficacy of these oncolytic viruses is a combination of lysis of the cells and indirect activation of anti-tumor immune responses. So here we have an oncolytic virus infecting the tumor cells. It's going to cause the cell to be lysed, but in addition, it is depending on what you put in the virus, it's going to attract immune cells like NK cells and antigen presenting cells that will work on the tumor as well. And in fact, we know that at the activation of local immune responses in a tumor can actually target tumors at distant sites. So that's called the abscopal effect, totally unanticipated, but you can have a local effect of your oncolytic virus, which will then be evident elsewhere in the body as well. And finally, of course, we use viruses for cancer immunotherapy, CAR T cells, which was FDA approved in 2017 for certain blood cancers. The idea here is that the tumor cell has specific antigens on it, but the T cells are not recognizing them. So we design a chimeric antigen receptor that will recognize this tumor antigen, and we harvest T cells from the patient and deliver this CAR gene, the chimeric antigen receptor, into the T cells via lentivirus vector. The T cells are then infused back in the patient and they home to the tumor and kill the tumor cells. So that's a very brief description of therapeutic viruses. And I just want to underscore again that these viruses have been made possible because of fundamental advances in virology, recombinant DNA, immunology, clinical science. And serendipity plays a huge role. There always has to be a balance between translational research and basic research. Forget about the Golden Fleece Awards. Let scientists pursue their curiosity, and things like this, therapeutic viruses, will come about. I guarantee it. All right. I hope you learned something there. Let's go back and take some questions. People get quiet. I know. Okay, let's see here. Where did we leave off with the questions? Okay. Last clinical update compared XBB vax, Novavax, older virus. How can they claim XBB is superior if previous asymptomatic infections were not determined? Well, yeah, it's part of the problem with saying it's superior. I don't agree with that, and I think I expressed my skepticism to Daniel, and he agreed as well, right? They're just the right experiments are not done, and we're just flying by the seat of our pants and telling people to get boosted, every 6 months. We've been complaining about this for a while. It just doesn't seem right. Kang writes, the last TWEVO on duplicate genes was interesting. Are there any natural examples of duplicate genes like those for antibiotic resistance in bacteria or duplicate genes in viruses? Yes, there are examples in both cases of gene duplications that, that lead to antibiotic resistance or viruses, often with anti-immune system effects in viruses. And I think Nels mentioned that, but more as well. Lee says, life should be full of serendipity. It is a gift and so grateful for serendipity. Life should be. Some people don't believe in it, right? They don't understand that it's part of everything. Okay, so Science 2 says the LV et al. Paper is being used by lab leakers to discredit two-jump theory regarding lineage A and B. Okay, so it's important for us to look at it, but this is BS. There are two lineages, there's no question, and they've been found within the market. So, the lab leakers, as I said last time, these market papers are devastating to the lab leaker hypothesis. So they just have to attack them with no basis. And notice, they attack them, but they provide no data for their own idea. There's zero data for a lab origin. Now, this paper I will have a look at, and we'll let you know what we think of it. Would an infection on top of an mRNA vaccination provide a more durable immunity? Yeah, that makes sense. So now it's hard to do the right study, right? Because so many people got vaccinated and naturally infected. And so comparing the longevity is going to be very hard, and people are continuing to get infected and boosted, right? So I don't know what to do. I think it's a real problem. Mark says, the hardest course I took as an undergrad was immunology with Elie Sarkars. Yeah, I didn't know him, but yes. Tough guy. Yep. Dr. Ehrlich wasted 605 experiments before 606. I'm not going to throw it because I'll just hit my screen. Well, they're not wasted because you had to make the progress to get there, right? So if you got the original 3 mRNA and nothing else, what do we get now? Well, Paul Offit would say if you're over 70, get the latest booster that they want you to. Otherwise, don't do anything because you're okay. You are able to respond. You probably made good T-cell immunity. The only thing is you don't know how long it's going to last, right? So I would say if you're over 70, look for the Novavax vaccine. That's what I would do. Mark Twain, history does not repeat itself, but it does rhyme. Gosh, I love this thing with these little bubbles, these little white, clear balls inside. They give it a nice texture. I'm going to pop this any minute. It's going to spill all over my keyboard. Jill said she felt cruddy after the mRNA switched to Novavax, 0 side effects. I mean, I would like to know if it's more durable, right? I've had multiple mRNAs. I never had any side effects. I got, after the first one, I had some fever for a few hours, but then the others, nothing. Lise writes, as a retired nurse, healthcare attorney, I agree some of the recent Supreme Court decisions, some of them are going to go bog down the courts even more. It's going to be a mess, folks, because if you just listen to the dissenting minority, these individuals are just espousing a conservative philosophy that's not compatible with life. It's ridiculous. I don't understand. White people couldn't work together. Anyway, we don't know anything about politics, but I just repeat what you guys are saying. Not crazy about holidays. Now, I'm not a big holiday fan. I don't know what you need. I like what I do. I don't need a holiday. Now, then you may say we have to celebrate certain things. Okay. Fine. So July 4th, we're celebrating our independence. But now we're going to screw it up. We're going to throw our democracy out the window, right? And have a king again, like what we got away from. So what's the point of July 4th? Martin Luther King Day is very good to celebrate. I agree. So I celebrate them in spirit, but I don't need to take time off personally. I will be doing my normal thing tomorrow here at the studio. And here we have another quote from Mark. Against stupidity, the very gods themselves contend in vain. Friedrich Schiller. Is it stupidity or ignorance, Mark? I don't know. I waffle between the two. Do you recommend going to my doctor if I think I have long COVID? Yeah, definitely. If you have persisting symptoms, you should definitely see a doctor. But you may find a doctor who doesn't believe in long COVID, so then you have to look around. I did an episode with a scientist at the Karolinska a few weeks ago on long COVID. I don't know when it's going to come out, but look for it for sure. I'm breathing. I'm breathing to calm down. The Euro trip spoiled you. Now you're back to the U.S. political dysfunction. It did spoil me. I mean, not that. I mean, Sweden has its problems, and I assume Finland does, but no one told me. My host in Sweden, Agnes, told me endlessly about Sweden's problems, but I didn't have a host in Finland, so I didn't hear about them. But Finland, is a huge country in land mass and 5.5 million people. What kind of problems could you have? Everybody's so spread out. But as I said last time, someone did say to me, I think it was Agnes, who said, the world looks up to the U.S. What the heck are you guys doing? And it's a very good question. I feel very uneasy these days. It's going to get worse as we head to the election. And so maybe I take asylum in Sweden. I don't want to give up on the U.S. Yeah, we go in cycles. Fascism is around the corner. I don't know. Democracies have not lasted forever, right? So maybe we're at the max. We're going to lose it, and then we're going to have to get it back. But that's beyond me. I won't be around. Watch Heather Cox Richardson. She says to do whatever you're best at. You're doing that, Vincent. Thank you. I learned what I was good at a long time ago. Teaching people about viruses, yeah. And I love it very much. What did Heather Cox's name came up? Was it last week? Oh, to get someone on to talk about the politicization of science, right? Onto Twiv. I don't know if she'd be the right person. Oh, it was an email to Twiv, actually. What do you think? Some of you may know Heather Cox Richardson. Would she be good to come on Twiv and talk about the politicization of science? Would she know that? Andy says, breathe in deeply through the nose, then out slowly through the mouth to the count of 10. Don't breathe in and out rapidly or you'll hyperventilate. Okay. Thank you. And FH says, keep repeating yourself. I learned in therapy that it takes many more reps of positives, or in this case, true science proportional to negatives. In this case, the rotten column in the New York Times. So rotten. People think because it was there, it must be right. And it's just a defective article, one sentence after the other. I do have lots of great virus toys. These are just a few of them. I have many others here. I have a lot at the incubator as well. Maybe we should all try deep breathing or a martini. Maybe, yeah. But Jill wants me to lead us all in a breathing exercise. Breathe in through your nose for 5 counts. Hold it for 5. Exhale through pursed lips for 10 counts. I can't do that. There will be silence, but everyone can do it on their own. You got us through the pandemic. Can you get us through this? I'm not a politician. I can't help. And when we venture into politics, we get criticized. The only thing I can do is try and point out the political fallacies with respect to science. And we had a letter on Twiv last week which said, Vincent, the lawmaker's job is to regulate science. Okay, fine. Then you should understand what science is. You don't even know what a gain of function experiment is. And you're going to regulate it. You're going to ban it outright, which is the dumbest thing. By the way, there is a question coming up from Kang. He says, these therapeutic viruses, are they gain of function? Of course they're gain of function. We're going to just ban all of them. Stupid frickin' Congress. They're going to ban all the therapeutic viruses because they're gain of function, and we're not going to have any monogenic therapy. We're not going to have any vaccines. We're not going to have any cancer oncotherapy. Okay, is that a good start? Good Lord. Okay. So I will do my best to deal with the science part. But there are a lot of other aspects of the crap that's going on that I listen to Heather. She gets me too upset. I can't even read her anymore because she gets me too upset of the gloom and doom. There is a place for educational toys, but accuracy is important. I think this one is pretty good with a few caveats that I pointed out, right? I would say the spike is the main thing. It doesn't have any threefold symmetry. But I don't know for the person on the street if that really matters or not, Bobby writes, I can't find any recent studies on IL-17 inhibitors. It's supposed to be promising as it's elevated in the cytokine storm. Yeah, the only one that Daniel ever talks about is IL-6, tocilizumab, right? Tosi. So there were trials. And in fact, in April, the results for one of these IL-17 inhibitors were announced. So these are monoclonal antibodies. IL-17 inhibitors were linked to a 36% increase in COVID occurrence. And Stelara was linked to a 37% increased risk compared to the controls. But this is these are people who were using IL-17 inhibitors for psoriasis. And so their use was linked to an increase in COVID. But I don't know, there most likely were. Here's a paper for 2020. If in Nature, is there a case for inhibiting IL-17 in COVID-19? Lots of inhibitors are available, as you saw for psoriasis there. And they argue that a clinical trial should be done. But, Daniel has not talked about it, so I don't know what the story is. I will have to ask Daniel. It's a good question. I'll put it on the sticky. Daniel, IL-17 inhibitors for COVID. Tomorrow is Thursday. It's going to be our clinical update. See, Daniel and I will record clinical update tomorrow, even though it's July 4th. Why? Because information must be disseminated. Immune-compromised female Aussie. My question, what is behind the recommendation that I have the inert shingles vax? Yeah, so the Shingrix is a protein. It does not infect you. And you are immunocompromised, so you don't want to take an infectious virus. And so that's why they recommend the protein-based vaccine, Shingrix. It's better than the other anyway, so you should get it, yeah. All right, I don't know what dark and stormy is. I'll look it up. Dark and stormy cocktail made with dark rum and ginger beer served over ice with lime. Doesn't sound bad. Someone want to make me one? Anger is about expectation and lack of control. Lower your expectations and accept how little control you have over others, and the anger will evaporate. Yeah, I know, but I'm living in a country where people are screwing it up. So I should just accept that? It's hard to do. What I do is distract myself by microbe TV type things, right? Like Livestream and all the other things we do. I mean, I agree with you. You're absolutely right. But it's not easy. Patricia says, Happy Independence Day 2024. Thank you very much. Just seeing that flag, I get very upset. You could make a Christmas ornament out of that glass virus. Yes, you could. This is so, but I don't want it to break. This is so beautiful. She did a great job blowing this. Oh my gosh. And she colored it a bit. And Mark says, if I have to match Vincent's glasses, that will be a lot of virus models. Well, you matched the ones I'm wearing tonight, Mark. We have brown and blue and have green. What else do I have here? I got lots of them. I have to line them up and show you them all sometime. Multiple protein-based jabs. Here in China, I've had 4 Sinovacs, attenuated jabs, 1 Vestvac, protein-based. Recently, I got 1 Novavax in the UK, but back in China, no chance of getting the second. Yeah, people are getting all kinds of things and also getting infected, most likely as well. Logotic says the beautiful glass virus reminds me of the glass flowers at Harvard. Yeah, that's right. This is so delicate, isn't it? It's beautiful. Thank you, Mark. Don't worry, says, is that where the name Lysol came from? No, I don't believe so. Mark Martin, did you know the origin of Lysol? Yeah, the name Lysol comes from combining the words lysosome and solvent. The original formula contained cresols, a phenol compound that could cause inflammation, burning, and even death. Before a formula change in 1952, early Lysol owners marketed it as a household cleanser, blah, blah, blah. All right, lysosome and solvent, created in 1889 to help end a cholera epidemic in Germany, but it had nothing to do with phages. I know that's what you were asking there. Bacteriophages can be used to convince bacteria to make antigens for vaccines. That's great. That's absolutely right. Yes. Craig says, using viruses like this still seems like Dexter, using a serial killer to your advantage. Yeah, that's the amazing thing. I find that you can repurpose viruses to do what you wish, right? Isn't that incredible? I mean, the HIV-based vectors, the lentivirus-based vectors, they've gone through several iterations. They're safe. It's really good. Betty is from Washington Heights. Oh my gosh, Betty. I spent so many years in Washington Heights, and I still have an appointment at Columbia University Medical Center, which is in Washington Heights. Our therapeutic virus is example of gain of function. So I ranted about this before. Yes, totally are gain of function. So if you want to ban gain of function, I got to take a deep breath, because why would anyone say ban gain of function without really understanding what it involves? I mean, this thing I told you today, this mini lecture, is so beneficial in so many ways. It's gain of function, and you're going to eliminate all of that. So obviously, they're not banning gain of function. They're going to have to throw it out. Just don't do it. Imagine the nonsense from anti-vaxxers if the COVID vaccine was made from hollowed out HIV. Yes, I'm sure somewhere someone was working on it, but you don't need to use a retrovirus, because you don't need persistent synthesis, just like you don't need AAV vectors to deliver antigens. You don't need them to be produced all the time. You don't want them to be produced all the time. Yeah. Yes, maybe Stephanie could explain Goff to politicians. Yeah, well, she could. Yeah, she should retitle her book "Gain of Function Saved My Husband." What is the title of it now? The Ultimate Killer, something like that. Which I don't think is a good title. The Perfect Killer, the Perfect Predator. No, it's not perfect. Nothing is perfect. Perfect is in the eye of the beholder. Andrew says a biologist was giving a talk to an audience of mathematicians. She asked, "You all know what a vector is?" Yes, they chorused back. Will says politicians need scientists to explain their work, but you need the right scientist. Congress invited Robert Redford to explain. Wrong. Yes, it was just the wrong person, because he's a Republican. He was appointed by Trump as head of CDC. The guy who said AIDS is only for gay people back in the 80s, right? This is someone who shouldn't even be allowed near the CDC. I mean, come on. Trump is saying he's going to defund any state that requires vaccination for school. What idiocy to even say this. I mean, I could see RFK Jr. saying this crap because he's not an elected official. But, oh my God, the former president and someone who wants to be president again, this is so ignorant. It's unbelievable. And you know what? I understand he's pandering to his voters. It's still ignorant as hell, and it ignores the lives of kids. So how do you get Congress to get the right people? I don't know. It's all wrapped up in politics itself. They picked Redford because he's on their side, and he says stupid things like the virus definitely came from a lab. God, could you say something more stupid? Oh, God. Good Lord. Yes, because they knew he would reinforce their incorrect preconceptions. They believe they want to blame China for the virus, and he will reinforce that. It's ridiculous. Good Lord. Are any of these therapeutic viruses used to treat cancer? Yeah, I guess you didn't. This was before that I talked about that. Yes. There are at least one license, right? The herpes virus for melanoma. There's another licensed in China as well. Yes. Newcastle disease virus is an oncolytic agent too. That wasn't in my list. Couldn't do them all. We don't want to be making a list of everything. The volume thing is, I have a volume indicator here and it's peaking, so turn up your end. But, I can show you my volume levels here. They're almost at max, so I don't know what else to do. Catherine used the Newcastle disease viruses against prostate and kidney cancer in vitro, and then I was introducing different interleukin genes to increase oncolytic activity. Yep, kind of like what we were saying too. Why do therapeutic viruses work for some people but not others? Everyone is different. When you infect 1,000 people with a virus, they all respond differently. Same thing with therapeutic viruses. The precise reasons, sometimes we think we know, right? Why some people get severe disease and others don't. But it's worth figuring out, right? I agree, but I wouldn't say that I know the answer to that. Yep. Yes, Pasteur said chance favors the prepared mind. That's correct. Serendipity is not the same now. They changed it to Sri Lankipti. Okay, and I don't get that either. Lisa wants to know if there's a microbiology board game. I don't know. Gut check is one. Okay, that's very cool. Oh, compound 606 was sarcasm given to us pharmacy students years ago. Let's see. Ehrlich's reagent, right? Well, I'm not going to read it all now, but so there weren't all those other compounds? Bobby writes, I've never had side effects from mRNA vaccines. I wonder if they're even working for me. Yes. It's hard to know, right? Did you ever get severely ill? Did you get COVID and was it mild? It could be the vaccine, but you don't know. This is part of the mystery of vaccination. The population as a whole does better, but individual people, we really don't know what's going on with you. But overall, they work. At a population level, we have reduced disease, right? So we use them. We got rid of polio in the U.S., paralytic polio in the U.S. by vaccination, no question about it. Yes, Will, you're right. Even young people benefit from boosters. They reduce the risk of long COVID. That's absolutely right. Absolutely. Thank you. People seem to disagree about it. Well, there have been a few recent trials that Daniel has talked about, and they support this notion that vaccination lowers the incidence of long COVID. I mean, it's not 100%. It's not 50% even. It's lower, but it's one thing that could be combined with other things to add up, right? As a molecular biologist, do you think some of these mRNA vaccines, which contain hundreds of thousands of DNA fragments, have incorporated itself into our genome? So they don't contain hundreds of thousands of DNA fragments. They contain hundreds of thousands of RNA fragments. And no, I don't believe they've incorporated themselves into our genome. I mean, some people have studied them in cells and culture and say that that can happen, but I don't believe that they have. I think their presence is transient, but even if they do incorporate into most of our cells that are turning over, it doesn't really matter. But I see no evidence that it is. No. As much as I respect and follow Paul Offit, he hardly ever addresses long COVID. A long time ago, I asked him about long COVID, and he said, well, what is it? It was a long time ago. So I think he has problems with the diagnosis, but I think we've moved beyond that also. So it's a thing. It's clearly a real thing. Yes. What's the latest edition for Principles of Virology? Fifth edition. The sixth is not going to be out for a couple of years, so you should get the fifth edition. Get the fifth, but don't plead the fifth. Mark says the problem is that the people who do bad things go home and have a good meal. Yeah, that's right. MN says people often ask on Reddit where a good place is to scream. In the woods, where nobody can hear you, right? Some people feel that screaming is good therapy. My own feeling on that is I wouldn't like to scream. It would shock me, right? I don't want to scream. So Kang says Heather should be a guest. Okay. Maybe folks, you write her and tell her she should be on Twiv. That helped for getting me onto Lex Friedman. Remember? All right. Les says the original German version of the quote, "Against stupidity, the gods themselves contend in vain," is that in German, which I will not say. Dummheit. Martha's in Charleston, South Carolina. Elizabeth says it's not that the politicians don't understand. Some of them know very well that they're lying. They see it as a way to gain power and control. Remember to vote, everyone. Take the power back. It's unfortunate, isn't it? I mean, just at the top, the very top, the president lies, right? Oh boy, gosh. But some of the things they say are so blatantly wrong, right? Let's defund a state who has vaccines, kids need to get vaccinated to go to school. Let's ban gain of function. I mean, for me, it's so laughably stupid. It's unbelievable. I don't get it. Do I ever reach out to the science or health reporters at the Washington Post? Near a pill may be receptive to your point of view. I don't know. What am I going to say? Reach out and say the virus came from nature, not in the lab, and don't believe any of it? I just don't know what to do. So what I do is just keep doing this. I do live streams. I do podcasts. I make videos. I teach. That's what I know to do. And maybe Fanit doesn't listen. I don't know. Catherine says she was excited to meet me at ASV. I'm sure there will be opportunities for sure. Mark has to make me a Hemingway Death in the Afternoon, absinthe and champagne. Yeah. Remember that bar we went to in Houston last summer where I asked the bartender if she could make a Manhattan with rum because they had a big rum collection. She did a good job. Tony wants to know about TNF alpha inhibitors and trials to prevent inflammation to treat rheumatoid arthritis. Apparently, it has anti-aging properties as well. I don't know anything about the anti-aging. I know it's in trials for RA, but I haven't kept up with it. I don't know. Can you explain protein-based vaccines for simpletons like me? Well, so a vaccine can be a virus, right? It could be a virus particle that you grow up and you inactivate it, or you attenuate it to make it able to replicate but not cause disease. Or in this case, this virus has spike proteins. You could purify the spike protein only, and it would be a protein-based vaccines. Now, Novavax, they don't actually purify it from the virus. They produce the protein in cells and purify it. But so you could have the whole virus as a vaccine, or you could just have a spike protein. So for Shingrix, it's the original attenuated vaccine was a whole virus. It was infectious. And the protein-based Shingrix vaccine is just one of the glycoproteins, one of the spikes on the virus particle protein produced in cells and purified. It's the Elton John of virology. Those different glasses. Thank you very much. Krishnan, good morning from Malaysia. Wow. You are far away. Welcome anyway. Although we are getting near the end here. Matters microbial number 18, the perfect predator, why phages rage. So Stephanie is really milking the what do you call these phrases? Phages rage, the perfect predator. I can tell you a lot of reasons it's not perfect, but I'm not. Mark is having champagne and guava juice. Mark, thank you for sticking with us. You usually go and cook dinner. Did you carry Paxlovid on your recent EU trip? No, I did not. I don't think we have learned much clinical immunology even with this pandemic. My disappointment is very deep on this score. Yeah, maybe not clinical. I think we learned a bit about induction of immune responses in lymph nodes and memory and so forth, but not clinical, yes. I agree about the 2/3. I will do more to share about TWIV. Also, yes, we'll PM you about stuff to help you with that BP issue. Have had the same trouble in spades. You're actually very chill overall. Yeah, I don't know about that. I tend to get very heated these days in talking about stuff. But yeah, I have the appearance of being chill, I suppose. Andrew says Sri Lanka used to be called Ceylon. Yes. And before that, Serendib, which is where we get the word serendipity. Oh, I didn't know that. Well, thank you, Andrew, for the information. I love it. And thank you for your support of science communication. If using multiple antivirals halts resistance, should we use multiple antibiotics for an infection? All right, so Mark Martin can answer this, but I say we don't because bacteria are DNA-based and their mutation rates are much lower, so you don't need to use multiple. Although you know, obviously you get antimicrobial resistance, but you don't want to use multiple because you might make that worse. What do you think, Mark? All right, Mark says, ask Daniel about his recent talk about ELISA-based over-the-counter tests for antibodies against SARS-CoV-2. So, Mark, here's my answer to that. What does a binding antibody mean? Right? And ELISA is going to tell you antibodies that bind to the spike. What does it mean? Is that what you need, or do you need neutralizing antibodies, or do you need, or maybe binding is good because they can have FC-mediated functions too. But I don't know. I'm not sure that they've done the studies to make a correlate, right? But I will ask him. What happened to movie night? Yes, we had said we're going to do movie night, which meant, so what we're going to do on a movie night? I don't remember. Talk about it, play clips, and discuss them. Remind me. I think it would be cool to play clips, and then we could chat about them. But I don't know if you're allowed to play clips like that on a live stream. I don't know. I'm one in a million. Thank you very much. I appreciate it. Appreciate your support. Thank you so much. All right, Mark's going for the dinner, so he's not going to answer about the antibiotics. But that's my thought. Mark at least wants to know what Mark's favorite cocktail is. I don't know. He left. But he mentioned a few, right? All right. Aerosmith, yeah, by Sinclair Lewis, used phage therapy for its plague cure. Yeah. Science writer Paul de Kreef did the research for the book. Yep, he did. He very much did that. Well, it's a good book. My mother used to teach it in her English class. I have a copy in the incubator with her handwriting in it. Well, folks, it looks like you have run out of questions. So let's wrap it up. Let's thank our moderators for tonight. That's fair use for education to cover movie clips. Good. Thank you, Ian. So we can do movie night. Pick a movie, I'll play some clips, and we can have a discussion. That would be fun. All right. Thanks to Les. Thanks to Andrew. Thanks to Tom for moderating tonight. I think that's it. Was Barb Mack here? I think she was here earlier. Barb Mack UK was here earlier. Now it's too late for her. Thank you all for being here, and thanks all of you for attending as well. So there are currently 123 of you and 97 likes. Hit the like button. The next office hours will be next Wednesday. That's July 11th, right? Is that correct? Yeah. No, not the 11th. Today's the 3rd, so that's going to be the 10th. July 10th, we will have our next office hours. Yep. So these movie nights, I guess you'd want to know what movie we're going to talk about beforehand, right? That's a good question. Renatus, thank you for your support of science communication. He writes, evidence shows that brain atrophy can result from prolonged isolation and reduced physical activity. Do studies linking COVID to brain atrophy consider this? Not that I'm aware of. I don't know. It's a good question. You know, the brain functions that are measured, well, there are control groups, but I don't know that they specifically look at isolation and reduced activity, which of course was a thing during the pandemic, right? All right, everybody. Thanks again for coming. Be safe. Have a good 4th of July, those of you who celebrate it, and be safe until next Wednesday. Thanks, everybody. Bye-bye.